In this lecture, we're going to be looking at all the different types of epithelial tissues that there are and how they are classified into those different types. So we'll start by looking at the classification, and there's two ways that we can classify an epithelial cell. The first is by its cell shape. There are three different cell shapes that we associate with epithelial cells. Squamous cells, and that's how you say that. It's tempting to want to call it squamous, but squamous cells are thin and flattened, or they may be kind of scale-like, where they lay over each other just a little bit. And they have, one way that you know that you're looking at a squamous cell is because the cell is so thin that it flattens out the nucleus inside the cell. So looking at them from the side, they almost have the appearance of like a fried egg, where there's a hump where the nucleus is, and again the nucleus will be kind of flattened out rather than being big and round. And then the rest of the cell is much thinner. The second shape is cuboidal, and cuboidal cells are roughly box-like. So if we think about that kind of a cell where the height, the width, and the depth are all roughly the same in a cuboidal shell and and because there's enough space inside the nucleus or inside the cell, the nucleus has a nice round shape generally, and it's usually located somewhere toward the center. And then the third shape is a columnar cell. These are taller cells, to me they always look like skyscraper buildings standing up tall. And because, again, there's enough space in them for the nucleus to be fully rounded, it's usually round, or the nucleus is round, and frequently they are located more down at the base layer. So sometimes they'll be squeezed up. This is a specific kind of columnar epithelium that we'll talk about later on. Okay, so three shapes that we can use to classify epithelial types: squamous, flat; cuboidal, somewhat box-like; and columnar, where it's taller than it is wide or deep. The second way that we can classify a epithelial type is by looking at how many layers there are in the epithelium. If it is a single layer, then we talk about it as a simple epithelium. And usually, not always, but usually, these are epithelia that are they're adapted for absorbing materials across the epithelial layer, or for filtering materials, moving fluid across, but not allowing the larger molecules to make it across that cell layer. The second kind of layering arrangement that we can have is referred to as stratified. With stratified epithelia, there are two or more layers of cells. So if a simple epithelium has just a single layer with the nuclei inside, then a stratified epithelium has multiple layers. So here's the basal layer, and then there's a second layer of cells upon that, and it can be a third, fourth, all the way up to, in some epithelia, there may be 50 or more layers of cells on top of each other. And frequently, stratification is there in order to allow for mechanical damage, so abrasion especially, as materials are moved across this cell surface. And then the third type of layering is referred to as pseudo-stratified. Pseudo means false. So if something is pseudo-stratified, that means that it appears to be stratified, but it's not really stratified. And this is frequently seen with mucous membranes or that have cilia or villi on them. And with pseudostratification, here's the basal layer, some of the cells, and they're often columnar cells, some of the cells are clearly, obviously attached to the bottom. Some may be a little shorter. And then other cells They are also attached at the bottom, but it appears, their attachment is very thin there at the bottom, and it creates this second row of nuclei because the nuclei are kind of squeezed out of being able to be attached at the bottom. So we get this appearance of two layers, even though there is only a single layer. So this image that I was showing here, this is a pseudostratified epithelium. Notice that all of the cells come down and have an attachment at the basal layer, but because some of them are squeezed so skinny we can't always see it, and because their nuclei might be squeezed up to a top layer, it gives the appearance of this stratification. So that's pseudo-stratification. So three arrangements. They can be simple or they can be stratified, and pseudo-stratified is actually a simple epithelium that has the appearance of stratification. So we can combine these three shapes and two different kind of stratifications, or stratification and no stratification, to make 6 different epithelial types, and then we'll add in 2 kind of wonky epithelial types. So with the simple epithelia, there's simple squamous, simple cuboidal, and simple columnar epithelium, and then of course the pseudostratified epithelia that we've already talked about. With stratified epithelium, there are stratified squamous, stratified cuboidal, and stratified columnar. And then there is also a type of stratified epithelium that's referred to as transitional epithelium. So let's take a look at all these different types. First are the simple squamous epithelia. These have a single layer of really flattened cells. The nuclei are also elongated or flattened. And they are very delicate and can be easily damaged, but they are found where it's really important to have very rapid diffusion or filtration of materials. So we find simple squamous epithelia in capillaries, in the lining of lymphatic vessels, on those serous linings, the body cavity linings, the serous membranes, and at the gas exchange surface in the lungs, where we want to be able to transfer a lot of gases very rapidly. And this layer here, where the dark nuclei, so these nuclei are the nuclei of this simple squamous epithelium. So it's a very thin layer. Directly underneath is connective tissue. That's not epithelium at all. And we've got a single layer right here at the top here of the lining. This is an arterial, a blood vessel. And so this simple squamous epithelium lines the blood vessel. A simple cuboidal epithelium, again, has a single layer, but this time the cells are nice and cuboidal shaped. So here you can make out the individual cells very easily with their big round nucleus centrally located. Sometimes there can be microvilli at the free surface or the apical surface of these cells, and generally these function to move materials either across the epithelium or, so moving materials from here and out, or there's a glandular secretion that occurs. And so some examples of simple cuboidal epithelium include the lining of the kidney tubules, where we are busy absorbing materials across these simple cuboidal cells, at the ovary surface, and in the ducts of exocrine glands, in the ducts of, say, sweat glands or oil glands, all the exocrine type glands. We'll talk more in a minute, or in the next lecture, about exocrine and endocrine glands and some of their differences. The third is a simple cuboidal epithelium. Simple columnar epithelium, and again a single layer of cells. Usually the cells are tall, and usually there's a basal spherical nucleus. So here's let's see if we can find one where we can. Okay, here's one. Sort of there. There's its nucleus. Here's another one. And there's its nucleus. And then we also have these goblet cells. They're the ones that stain blue. goblet cells produce mucus, while the rest of the cells are there forming the lining. You can also see the microvilli. That little layer right there is all microvilli. They're helping to increase absorption in the intestinal tract. So a single layer of tall cells, generally the nuclei are basal, so you can see that they're kind of here. Most of them are here in this lower portion of the cell. And they can have cilia or microvilli on the free surface, there at the apical surface. And frequently they're interspersed with these mucus-producing goblet cells. And their job is to move materials through tubes. So we're trying to get material to move across the surface of the cell, and then across the epithelium to bring materials through the cells and into the body fluids on the opposite side of the epithelium. And so we'll see a simple columnar epithelia at the lining of the ovary tubes, the ovarian tubes, where the cilia help to wave. They wave, and they push the egg through the uterine or the ovarian tubes. You know, that shouldn't be ovarian, that should be a uterine tube. I apologize. So through the uterine tubes, and then also in the lining of the digestive tract, where we're absorbing nutrients into the body fluids and kind of sliding the food material along on this mucus layer that the cells produce. So simple columnar epithelium. And then last of all for the simple epithelia is the pseudostratified epithelium. Again, this is the false stratified. It's not truly stratified because all of the cells attach at the basement membrane, even though many of them may not have their apical surface, or have an apical surface there at the free surface of the epithelium. It appears that the nuclei are at two different levels, which gives it that stratification. Most pseudostratified epithelia have cilia up here at the apical surface, and the goblet cells are interspersed, delivering mucus there to the surface. And the mucus will pick up like bits of dust that might be in the air that we're breathing, because these pseudostratified epithelia are, this is the lining of the trachea and the airways in the lungs. So the mucus will kind of glom onto all of the particles in the air that we're breathing, and then the cilia will wave that mucus up the trachea, up into the pharynx, where we can then swallow the mucus plus all the impurities in the air that we're breathing. So those are the simple epithelia. Now let's take a look at the stratified epithelia. Stratified squamous epithelia, they are found in any area that must maintain mechanical or integrity against mechanical stress or chemical stress or even drying stress. So what we're looking here at in this picture is a stratified squamous epithelium from the esophagus, where we can see all the layers of cells. Down here is the basal cell layer. Here's the basement membrane. And then these are the cells. Notice at the basal layer, they are actually cuboidal cells, but the further they get away from that basal layer, the more flattened the cells become until they assume this very characteristic flattened squamous shape. So with stratified epithelia, like stratified squamous epithelia, we're constantly replacing cells that are lost at the apical surface. Up here at the free surface, you can see how the cells will flake away, and in order to keep this thickness of epithelium, then we've got to replace those cells. Replacement begins down here at the basal layer. These are stem cells here, and they will divide constantly and push the newly divided cell up as more cells are placed underneath it. And so we're constantly pushing these layers upward, losing them away from the epithelial surface here at the apical surface, and then, sorry about that, replacing them underneath at the basal layer. It's almost like the grass in your yard. If you mow your grass, you know that you got to mow it every week, because you trim off the upper side, now this is gone, and you have this raggedy edge. It doesn't grow again from here. It's growing from way down here near where the roots. This is the area where it grows from. So new cells are constantly being added there at the bottom layer. So within a week, it's back up again, and we come and mow it off once more. It's the same with these stratified squamous epithelia. They're constantly being replaced from down at the basal layer, constantly being removed from up at the apical surface. So some stratified squamous epithelia may be keratinized. That means that the cells up here at the toward the apical surface are stuffed with keratin. This is a protein. It's a waterproof, white-colored protein. And as the cells are pushed toward the apical surface, those cells will flatten, harden. With all of this keratin that's put in there, we'll start losing organelles because there's no room for organelles with all the keratin in there, and the cells will eventually die. And so these keratinized cells then form this dry, tough, waterproof covering that helps to protect the underlying cells and tissues. So the skin is an example of a keratinized stratified squamous epithelium. Whereas the other places where we need all that mechanical protection, but we need the cells to also be moist and flexible and alive, in those stratified squamous epithelia, the cells may be non-keratinized. The cells remain alive and are soft and moist, but they can still peel away at the apical surface. And so the linings of the mouth and esophagus, the lining of the vagina and the anal canal, all of these places have non-keratinized but stratified squamous epithelia so that they can replace themselves as materials scrape through these areas. Okay, so that's the stratified squamous epithelia. Stratified cuboidal epithelia are usually only 2 to 3 layers thick. That's because they don't have access to nutrients easily, and they may be found lining the lumen. Again, that's the hollow space within a cavity within a hollow organ. That area encircled is the lumen. They're relatively rare in the body, but we find them in the pancreas, in the lining of some salivary gland ducts, and the ducts of mammary and sweat glands. So not a real abundant tissue type. Stratified columnar epithelia are also relatively rare in the body, and usually found with only 2 or 3 layers. Again, because the cells are so deep, it's difficult to get nutrients up to the apical surface. They also line the lumen of a structure, and are found in the pharynx and in parts of the urethra in males. This is the urethra. Remember, well, you don't know this yet. The urethra is the tube that carries urine from the bladder to the external surface of the body, and so that's a place where we find stratified columnar epithelia. All right. I think that, oh, no, one last more, sorry. Here's the last slide. And this is the transitional epithelia. Transitional epithelia are stratified, and we can see here that they're stratified. The basement membrane is down here. Let's see, did I miss it? Yeah, right through there. Down below, this is connective tissue, and so these are the nuclei of the fibroblasts that are maintaining the connective tissue. But up above is the transitional epithelium. Notice cuboidal, and when they're relaxed, the cells tend to maintain this cuboidal shape. Get down to where the stem cells are. Here are the stem cell layers. So they have a relaxed cuboidal shape when they have a cuboidal shape when they're relaxed, but when the hollow organ that they're lining is stretched, perhaps because it's filled with urine, and that's really the only place that we find transitional epithelia, are in the urinary bladder, in the ureters of the urinary system, and even in parts of the urethra. But as they stretch, these cuboidal cells, I'm going to draw. I'm trying to draw. Okay, it's not going to let me draw. Come on. Let's do this. There we are. Okay, now I can draw. Here's the the basement membrane. Here are the cuboidal cells. Again, just a few layers. And up here at the surface, they have kind of rounded edges. This is what will help you to recognize. Notice how that upper layer is rounded. When we stretch out say the urinary bladder, the cells get stretched too, and they become much more flattened looking. So they transition from being cuboidal to being more squamous as the urinary bladder fills with urine and stretches to accommodate all that urine. Cells go from being, or they transition from being cuboidal to being more squamous. So we're going to see slides, the slides that you'll be looking at in lab, they're all going to be relaxed transitional epithelia. So you should be looking for those rounded cells there at the apical surface. Okay, so that does it. We've looked at all 8 types of epithelia that I want you to be able to recognize. If you have any questions, let's talk about it in lab or send me an email.